Gabby, looking forward to hearing.
Awesome. Thanks, Dr. Farrell. Let me just. Sorry, everyone. Just give me 1 second. Perfect. There we are. Are we able to see okay, Danny, on Zoom? Yep. All right, awesome. Hello, everyone. Thank you so much for making it out today in person as well as online. My name is Gabby, and for my second master's seminar today, I will be discussing my project titled Assessing the Clinical Relevance of BRCA1 BRCT Domain Variants of Uncertain Significance. For this talk, I'll start off with some introduction and background for my project, followed by my hypothesis and rationale, experimental aims and results, my limitations and next steps, as well as future directions and the overall significance of this work. So starting off with some background, breast cancer is one of the most common malignancies and one of the leading causes of cancer-related death in women worldwide. Ovarian cancer, while less common than breast cancer, is 3 times as lethal. Most breast and ovarian cancer cases are considered sporadic. However, about 5 to 10% of breast and around 20% of ovarian cancer cases are linked to inherited mutations in cancer susceptibility genes. Hereditary breast and ovarian cancer syndrome is an autosomal dominant inherited disorder most commonly associated with germline mutations in the tumor suppressor genes BRCA1 and BRCA2. Those with HBOC have a significantly elevated risk of developing breast and ovarian cancer in their lifetimes, as well to a lesser extent, prostate, pancreatic, and melanoma. Given the causative link between BRCA1 and 2 mutations in HBOC, genetic testing has become widely implemented to identify carriers of pathogenic BRCA variants. Although genetic counseling is essential for determining which individuals would benefit most from genetic testing, BRCA1 and 2 screening is generally recommended for women who have a family history of breast or ovarian cancer, are of Ashkenazi or Eastern European Jewish descent, have a known BRCA pathogenic mutation in the family, or have personal history of breast or ovarian cancer. The outcomes of clinical genetic testing greatly impact the next steps for patients. A positive test, meaning a pathogenic variant has been identified, warrants the enhanced surveillance through mammographies and pelvic ultrasounds, as well as the investigation into risk-reducing strategies, such as prophylactic surgeries, as well as chemopreventative treatments. Those are for healthy carriers. However, for cancer patients, this warrants the investigation into their eligibility for targeted therapeutics, such as PARP inhibitors. Additionally, genetic counseling and reflex testing for respective family members who may also have a pathogenic variant is then warranted. Contrarily, a negative test results in the circumvention of any unnecessary interventions and helps with reducing anxieties about cancer, financial strains, as well as reproductive issues. Although genetic testing has greatly advanced the field of precision medicine, its widespread implementation, as well as commercialization, has resulted in the discovery of thousands of variants of uncertain significance, or VUS. VUSs are DNA sequence alterations that have an unknown effect on protein or gene function and subsequently disease risk. Approximately 20% of BRCA1 and 2 genetic screens results or report a VUS, and about 13% of all BRCA1 and 2 variants reported through these screens are reported as VUS. When looking at the BRCA1 gene, approximately half of its 14,000 germline mutations that are currently documented on the ClinVar variant classification database are reported as VUS, not provided, or have conflicting classifications, meaning that there were multiple submitters asserting different classifications. Missense variants, which are coding alterations that have a substitute of an amino acid residue, are a class of variants that are particularly challenging to determine clinical relevance. And this is a predominant challenge in BRCA1, as demonstrated through this pie chart on the right. The majority of documented missense variants in this gene have some level of uncertain significance. VUSs, they pose a critical clinical challenge because in the common scenario in which a VUS is identified, clinicians are unable to effectively interpret genetic testing results, leading to uncertainty about next steps for patients. So in the context of BRCA1, a VUS results in uncertainty about prophylactic treatments or risk-reducing strategies that patients can take, and also leads to patient-induced anxiety. Additionally, the identification of a VUS does not grant eligibility for cascade testing for family members who may also be at risk. As alluded to in the previous slide, my focus lies in the BRCA1 gene. BRCA1 is a pleiotropic tumor suppressor gene involved in various interconnecting cellular pathways that ultimately promote genomic and cellular stability. One of BRCA1's functions that I'd like to highlight is its role in DNA double-stranded break repair. So through protein-protein interactions with select binding partners, BRCA1 forms macrocomplexes that are crucial mediators of the DNA damage response. Sensors that detect DNA damage interact with these complexes, and these complexes subsequently activate cell cycle checkpoints, as well as recruit effectors to sites of DNA damage to repair them through homologous recombination. Thus, when a pathogenic mutation compromises the normal functioning of BRCA1, alternative processes like non-homologous end joining are activated, and these error-prone compensatory mechanisms result in increased genomic instability and subsequently promote tumor progression and tumor genesis. The BRCA1 protein consists of several functionally distinct domains. The N-terminal ring domain functions primarily as an E3 ubiquitin ligase that regulates various post-translational cellular processes, including protein degradation, DNA repair, and cell cycle regulation. The middle region, I'm just gonna take this off. I hope everyone can hear me okay. All right, thank you. The middle region, specifically exons 11 through 13, consists of several functional components, including a nuclear localization sequence, transcriptional regulator binding sites, a coiled coil domain involved in homologous recombination, as well as a serine cluster domain that's involved in BRCA1 recruitment to sites of DNA damage. And on the C-terminal end, we have the BRCT complex, or the BRCT protein domain, I should say, consisting of the BRCT tandem repeats with a linker. This domain is involved in DNA repair through its interactions with key phosphopeptide binding partners, Abraxas, CTIP, and BOC1. BRCT interacts with these proteins separately to create macrocomplexes that have distinct and overlapping functions in homologous recombination. The Abraxas complex, for example, is involved in BRCA1 recruitment to sites of DNA damage. The CTIP complex is involved in DNA resection. And finally, the BOC complex has been shown in cell cycle regulation, as well as effector functions in DNA repair. Keeping the structural organization of BRCA1 in mind, the next question is where the thousands of BRCA1 variants, specifically missense variants, occur within this gene. These histograms, created by previous Davy Lab student Kamran Islam, demonstrate that pathogenic variants localize primarily to the N-terminal ring and C-terminal BRCT domains, while benign variants appear to distribute relatively evenly throughout the gene. VUSs, on the other hand, they follow a very similar trend to pathogenic variants, in which above the background amount of variants, there are clusterings in the N-terminal ring and C-terminal BRC domains. Given these trends in these histograms, as well as its established role in DNA repair, the BRCT domain became a topic of interest for me, as I expect a larger proportion of pathogenic VUSs to localize to this domain. Matt, who is another master's student in our lab, is focused on the ring domain for similar reasons. That's not. So with clinical labs increasingly detecting new sequence variants through genetic testing, the American College of Medical Genetics and Genomics have established detailed variant classification guidelines to streamline the variant interpretation process. So for a variant to achieve either a benign or pathogenic classification, they must attain or they must meet certain criteria using different types of evidence. So as you can see, some types of evidence are stronger than others. However, evidence types from supporting to very strong can be combined to achieve a certain classification. I'd also like to note that the criteria highlighted in the black boxes are types of evidence that I'll be delving into further in this presentation. And as well, this is more so a representative figure, just showing some of the criteria within the guidelines, and I've excluded some of the criteria that aren't as pertinent to my project. Although these guidelines work to reduce the number of variants that are reported as causative of disease without having sufficient evidence, this very stringent model has also resulted in a larger proportion of variants being classified as VUS. Missense variants account for the large majority of VUS in BRCA1, as there is often insufficient evidence to achieve a benign or pathogenic classification due to the very low minor allele frequency of individual variants. And this results in a limited application of segregation and population-based analyses, which are typically used to interpret sequence variants. Pedigree analysis is one criterion that can achieve up to strong evidence and is a traditional gold standard method for sequence interpretation. This approach requires tracking the health of carrier and non-carrier family members to assess whether a variant segregates with disease. While this method is well proven, it requires multiple families, each with enough living family members, as well as years of follow-up to obtain enough a high enough penetrance for a VUS with disease. Currently, under Ontario guidelines, the identification of a BRCA1 VUS in an individual does not allow for reflex testing for at-risk family members. However, it can be assumed that the establishment of a BRCA1 pedigree analysis database can have long-term payout in the improvement of our understanding and our ability to interpret VUSs. And this is a future goal that I'll touch on later in our presentation. So this leads to the rationale for my project. Would it be possible to confidently interpret the pathogenicity of BRCA1 VUSs without pedigree analyses? Sorry about that, everyone. There we are. Sorry about that, everyone. So just to recap again, would it be possible to confidently interpret the pathogenicity of BRCA1 VUSs without the need for pedigree analyses or other labor-intensive methods? In silico computational tools are already used as supporting evidence for classification, but what if there was a superior BRCA1 specific computational tool that would be able to predict VUS pathogenicity, as well as guide the decisions into which VUSs we should conduct additional analyses on to obtain even stronger evidence? As I mentioned in previous slides, BRCA1 is a pleiotropic gene that has multiple functional domains. So an approach for creating a superior BRCA1 predictive tool is to tailor a tool for a functional domain using a custom selection of in silico tools. And that leads to my hypothesis for the project, that a BRCA1 BRCT specific missense variant classifier trained on known consequence variants can be used to predict VUS pathogenicity, and these predictions can then be confirmed with functional assays. So I have three aims for this project. First being to build a domain-specific classifier using a combination of select in silico tools. Second, to assess the impact of select VUSs on BRCT binding ability using a pull-down assay. And finally, investigating the feasibility of a PARP inhibitor sensitivity assay to assess VUS therapeutic relevance. One future aim I'd like to note is to integrate the results from this project into future pedigree analyses on select VUSs. Starting with our first aim, which is building the classifier, the first step was data collection. So over 1,000 variants, missense variants spanning the BRCT domain were collected from the ClinVar classification database, including known pathogenic and benign variants, as well as VUSs, variants with conflicting interpretations, as well as those that were not provided. Then approximately 50 in silico tools were combined or compiled from multiple databases and sites, most notably from the variant effect predictor tool. In silico, an expression derived from other experimental terms such as in vivo and in vitro refers to a diverse range of computational tools that analyze, model, and predict biological processes. So the in silico tools that I used predict variant pathogenicity based on different aspects of sequence conservation, such as the biochemical or structural impact of a residue substitution or evolutionary sequence conservation. But the majority of the tools that I used were machine learning based, in which they had algorithms that were trained on large data sets of known consequence variants. This is just a representative figure showing just some of the recognizable tools that I used, as well as the categories that they fell under. I then applied each tool to known pathogenic and benign BRCT variants to obtain their respective pathogenicity scores. This is also a representative figure showing just a fraction of the variants I assessed, as well as the scores that they obtained for certain tools. I'd like to note here that each tool uses their own scale to score pathogenicity, but I formatted the sheet such that green cells represent more benign scores and red cells represent more pathogenic scores. Hopefully you can see the separation in color already between pathogenic and benign variants. However, as you can visually see as well, there are some tools that perform and discriminate better than others. To select the best tools to incorporate into my classifier, being the tools that most accurately discriminate known pathogenic and benign variants, I first min-max standardized my data to a 0 through 1 scale, split my data into a training and validation set, containing labeled pathogenic and benign variants ranging the BRCT domain, and finally ran this data through the program Molecular Feast. Molecular Feast, or MFeast, stands for Molecular Feature Selection Tool and is a machine learning-based application that has been developed by Dr. Catherine Trishkin as well as the Renwick Lab. MFeast uses an ensemble-type feature selection algorithm, meaning that it utilizes multiple types of feature selection algorithms and utilizes a greedy algorithm to combine the results of the different methods to rank features. Feature selection is the process of isolating, identifying the most consistent, non-redundant, and important features to use in model building. So in the context of my project, MFeast was used to identify and rank the most important in silico tools to integrate into my classifier. MFeast feature selection ranked tools based on their discriminatory ability. So I've included a table on the left side here, showing just a fraction of the top ranked in silico tools from the algorithm. In the middle is a predictive plot showing the discriminatory ability of the ranked tools going down the list, and the plot provides a visual estimation of how many tools I'll be integrating into my classifier. And finally, I've included a t-SNE plot here generated using the top 10 tools, and this figure visualizes the separation of benign and pathogenic into two loose groups. Of note, and this will be elaborated more on in Matt's presentation, there was variation in the ranking of tools for the BRCT domain and when this was done with the ring domain. So this further suggests that there's benefit of building these models separately. So with ranked tools established, I next move on to building the classifier using the MATLAB Classification Learner app. So Classification Learner is an interface on MATLAB that enables the building and training of multiple types of models on any given labeled data set. So starting with the same training data that I did for MFeast, I incrementally trained models on Classification Learner, each time integrating one more tool from the ranked list. And I did this to identify which tool combination would result in the most accurate model on the training and validation sets without introducing any redundancy by using more tools than needed. So through this method, I found that an ensemble subspace KNN model trained on the top 9 ranked in silico tools produced the highest training and validation accuracy with 91 and 88% accuracy respectively. KNN, or K nearest neighbors, is a type of supervised learning algorithm that works on a similarity measure. So it predicts the correct class for test data based on the calculation of the distance between a test data point and the training data point. I've included a confusion matrix here, just demonstrating the performance of this model, as well as an ROC curve, demonstrating the performance of the model at different classification thresholds. I next wanted to compare this new model to a growing topic in the field of variant interpretation, and that's AI protein language models, such as Google DeepMind's latest release, AlphaMissense. So AlphaMissense is an unsupervised learning algorithm that incorporates structural context with weak labels from population frequency data to predict the impact of every possible missense variant in the human proteome. So although AlphaMissense can help on a broad scale to provide insight into the functional consequences of genetic variation, when compared to the KNN's ability to discriminate and predict BRCT variants, the KNN model consistently performed with higher accuracy on test sets compared to AlphaMissense. Although the accuracy is only slightly better numerically, when looking at the model's ability to predict pathogenic variants, AlphaMissense misclassified 6 more pathogenic variants as benign compared to the KNN model. And although this may not seem as a lot, in the context of BRCA1, a pathogenic variant being incorrectly classified as benign can be detrimental. The next step of this workflow was to apply this KNN model to our BRCT VUSs. Scores for all VUSs were obtained from the top 9 in silico tools, standardized, and then run through this model. The KNN model then scored the variants on a 0 through 1 scale, with 0 being benign and 1 being pathogenic. The histogram in the middle depicts the distribution of the scores that the model gave the VUSs. Finally, I selected VUSs of interest, being 6 likely pathogenic variants that either scored a 1 or slightly below a 1, as well as 7 likely benign VUSs scoring either a 0 or just above a 0. So as a summary of aim 1 for this project, I created a BRCT specific KNN subspace classifier using select in silico tools. And this classifier demonstrated to have a higher predictive accuracy on an independent data set compared to other in silico tools and AI models. This classifier was then used to predict the pathogenicity of all missense BRCT variants in the ClinVar classification database. And this allowed me to rank BRCT VUSs based on their pathogenicity score, and then subsequently select a subset of VUSs for further analysis. So the next aim of this project is confirming the results of this classifier by assessing how these variants of interest impact BRCT functionality, specifically how these VUSs impact BRCT's ability to bind with its phosphoprotein binding partners, Abraxas, CTIP, and BUC1. So this is a video that I generated on PyMol demonstrating the interactions that BRCT, which is colored in blue, makes with its 3 binding partners, color-coded to the legend here at the bottom. So as the phosphoproteins cycle through, I'd like to highlight one commonality between all 3 phosphoproteins, and that's that BRCT recognizes each of these proteins by their phosphoserine XX phenylalanine motif, with the 2 X's representing any other amino acid. So if you follow my cursor here, hopefully it won't stutter too much, you can see that distinguishable ring for the phenylalanine, as well as that phosphoserine there. And this motif, it functions as a 2-anchor interaction mechanism in which the binding partner interacts with both BRCT repeats separately. A key thing to note here is how among all 3 binding partners, the phenylalanine and the phosphoserine localize and bind relatively in the same position within the BRCT binding pocket. This being said, there still are visible differences, visible and biochemical differences in how the proteins bind and elicit their respective mechanisms. I would also like to highlight the red and green colored amino acids located in the BRCT protein structure. So these residue positions are the positions of the VUSs that I selected from my aim 1. The first thing I'd like to note here is that their location within the BRCT protein. Let me see if I can restart the video. You can see that the benign variants are located in regions more so on the outskirts of BRCA or the BRCT protein, and not in any structural components or any binding sites of BRCT. Contrarily, when we're looking at the suspected pathogenic VUSs, they're either directly in the binding pocket of BRCT, or they are in structural components such as alpha helical coils or hydrophobic interfaces. And these are trends I'll be delving into further in the presentation. So to assay each variant's ability assay each variant's impact on BRCT binding ability, I employed a pull-down functional assay. So at the beginning of the workflow, I designed DNA fragments consisting of the BRCT sequence, an N-terminal flag tag, as well as there's corresponding nucleotide substitution. I also created fragments for the wild type as well as the pathogenic control. So these fragments were topo-cloned and plasmid purified, gel extracted, and finally ligated into the pTra2 mammalian expression vector. This vector was transfected into 293 T cells, and following a 2-day incubation, they were harvested. Cells were lysed and then separated into two groups, a whole cell lysate as well as a BRCT protein purified lysate. So this purified protein was obtained through a co-immunoprecipitation reaction using magnetic anti-flag beads. So these magnetic beads had anti-flag antibodies on them that specifically bound to the flag tag, a part of the BRCT construct. And so the magnetic beads isolated the BRCT and eluted them off into a final product of just our BRCT protein construct, as well as whatever binding partners it bound to. Western blots were then conducted, probing for flag and the 3 binding proteins. And then binding was quantified by normalizing the band signals from bound proteins back to the signal exhibited by the wild type control. These normalized signals were then divided by the corresponding normalized flag signal to obtain binding activity relative to the wild type. So let's begin with some results for BRCT's ability to bind with Abraxas. So starting with the suspected pathogenic variants, 4 out of the 6 variants demonstrated significant loss of binding compared to the wild type. And a key thing to note here is that the arginine to proline as well as phenylalanine to serine demonstrated even more binding loss compared to the pathogenic control, which is quite interesting. As for the other 3 variants, the glycine to valine variant does show a trend of loss of binding. However, the last 2 variants here, tryptophan to lysine as well as phenylalanine to serine, demonstrate retained binding in comparison to the wild type. As for the benign variants, based on the 2 replicates that we have so far, binding is retained in relation to the wild type. Moving on to CTIP binding ability, 5 out of the 6 suspected pathogenic variants show significantly lower binding ability compared to the wild type. However, that same phenylalanine to serine variant demonstrates retained binding. Benign variants appear to follow a similar trend as the last slide, where they have retained or even have an over-retention of binding to CTIP in comparison to the wild type. I will note here, when we're looking at the bar graphs and compare them back to the actual Westerns I've included at the top, I want to elaborate a little bit on why it appears that the banding is quite similar, especially for the pathogenic variants, and why they look a little bit different on the bar graphs. And that's because when we normalize them back to the flag, there was a lot less flag, for example, in the phenylalanine to serine. So when we divide the signal of the binding protein to a flag signal that's a lot less than the other ones, that results in that increased relative binding activity. I'm still in the midst of obtaining results for the Bach binding efficiency for the pathogenic and benign VUSs. However, the results from the Abraxas and CTIP binding assay generally align with the predictions made by the KNN model. However, it also left me to question why certain suspected pathogenic variants, such as the phenylalanine to serine variant, demonstrated binding retention despite being predicted as deleterious by the KNN model. Is it possible that there's another mechanism of loss of function that is outside of just losing binding activity? So that question actually goes hand in hand with another issue that I ran into with this assay, and that was the inconsistency of flag banding in my Western blots across the variants. What was initially assumed as variation in the amount of protein that was added to the wells became a very strong trend in which the majority of suspected pathogenic variants, with the outlier of arginine to proline, interestingly enough, demonstrating significantly less flag banding than the wild type. Benign variants, on the other hand, demonstrated consistent flag in relation to the wild type. And this resulted in further analysis into what was going on here. So we had two assays that we conducted. First was assessing whether there were differences in the transfection efficiency of the P tray vector into the 293 T cells. So I labeled wild type and pathogenic P tray BRCT vector with a Sci-Fi fluorophore, transfected them into 293 T cells, and conducted flow cytometry to visualize the percentage of cells that successfully took up the vector. And as you can see through these histograms here, the transfection efficiency was quite consistent across wild type and pathogenic constructs. So the next test that we did was a BRCA specific co-IP. So instead of using the anti-flag antibody to pull down our constructs, we use an anti-BRCT antibody to pull them down to assess whether it was a problem with the magnetic beads that were, for whatever reason, unable to pull down the pathogenic constructs. However, we're seeing a very similar trend here where the wild type constructs were able to be pulled down by this new antibody. However, the pathogenic construct still wasn't able to be pulled down as effectively. So given that the differing flag levels was not a result of transfection efficiency or pull down efficiency across the wild type and pathogenic constructs, I next investigated potential structural or biochemical explanations for this. One trend that I found particularly interesting was how the 5 pathogenic VUSs, as well as the pathogenic control, showed a lot less flag banding, but arginine's flag banding seems to have been retained despite it being one of the strongest variants to show tendencies of pathogenic activity through its ability or through its loss of binding to Abraxas and CTIP. So that's what brought me back to looking at the structure of BRCT and its binding partners again. So as you can see, arginine 1699, that residue position right there, is in direct contact with CTIP, but also with its other binding partners as well. It makes several hydrogen bonds with the phenylalanine, which is the phenylalanine, a part of the phosphoserine phenylalanine motif. Thus, it's acting as a strong anchor point for binding. And when it's substituted with the white proline, which will cycle back, those interchain bonds are visibly disrupted. So it's evident that an amino acid change in this position would directly impact BRCT's binding ability. However, if you look at the other suspected pathogenic variants, they're either located in alpha helical structures or the hydrophobic interfaces and not directly in contact with the binding partners. Phenylalanine to serine, the one right behind arginine 1699, is an interesting case such that it's involved in an alpha helix as well, to a lesser extent, binding to a phosphoprotein. So this observation suggests that arginine 1699 proline directly impacts BRCT's ability to bind. But as demonstrated through its elevated flag levels, as well as its residue location not being within any structural elements of the protein, it does not detrimentally impact protein folding. The variants in the helical structures and the hydrophobic interfaces, on the other hand, though, likely impact key structural components of BRCT, suggesting that they impact native protein folding and stability, and thus result in the protein no longer to fold properly, resulting in degradation and loss of function. And this is an assumption or a hypothesis into why we're seeing less of this flag in our Westerns. So as a summary for AIM-2, the pull-down assay results generally align with the predictions from the KNN model. There were several outcomes observed for pathogenic VUSs, where they either demonstrated unstable protein folding, a loss of binding activity, or a combination of the both. Each of these outcomes result in less effective BRCT domains, though, in which overall binding to phosphoproteins is reduced compared to the wild types. Benign VUSs, on the other hand, demonstrated stable folding and retained binding ability in comparison to the wild type control. So that's what brings me back to this last aim, which is still underway, and that's assessing the feasibility of a PARP inhibitor sensitivity assay to assess VUS therapeutic relevance. So PARP inhibitors are clinically used as targeted therapeutics that employ a principle called synthetic lethality to specifically target BRCA1 mutated cancer cells. They do this by inhibiting PARP, which is an important protein in DNA single-stranded break repair, inducing single straights that manifest into double-stranded breaks during replication. So cells that have deficient homologous recombination abilities, such as those that are deficient in BRCA1 or have mutated BRCA1, are unable to repair their DNA and are preferentially killed. So the foundation of this aim is to investigate whether the introduction of wild-type BRCA1 into a BRCA1 mutated cell line will restore cells' ability to perform homologous recombination and remain viable when treated with PARP inhibitors. If so, this method can then be applied to VUSs in order to assess whether BRCA1 with a certain VUS can be introduced into a mutant cell line and then can be assessed by its ability to either rescue the cell's ability to remain viable when treated with PARP inhibitors or actually sensitize cells to PARP inhibitors. So the workflow for this aim is introducing an expression vector with full-length wild-type BRCA1 into MDA-MB436 cells, which is a BRCA1 mutated cell line, treating them with increasing doses of olaparib, which is an established PARP inhibitor, and finally conducting a clonogenic assay, which will be used to assess the survival of these cells in comparison to the control, which has no BRCA1 incorporated. So on a broad scale, being able to interpret whether a VUS can act as a predictive biomarker for a cancer's sensitivity to target therapeutics like PARP inhibitors would show direct benefit in the clinical setting. So rounding out this presentation, I'd like to address some of the limitations and next steps for this project. For aim 1, as already outlined in the ACMG guidelines, computational tools should only be used as supporting evidence for classification. Although the KNN classifier has shown to be more accurate at predicting variants in BRCA1, BRCT domain compared to in silico tools on their own, as well as newer AI tools, this should Okay, perfect. Are you able to? Okay, awesome. So although the KNN classifier demonstrated to be more accurate compared to in silico tools on their own, as well as AI classifiers, this tool should only be used as supporting evidence. In terms of aim 2, despite the gathering of information about the variant's impact on binding of proteins as well as protein stability, there's still a lot of ambiguity in these pull-down results. Firstly, despite how I framed our current knowledge of BRCA1's multiple roles in tumor suppression are still not fully elucidated. Thus, it cannot be assumed that the loss of one aspect of its DNA repair mechanisms, in this case being its ability to bind to phosphoproteins, necessarily debilitates its entire tumor suppressive activity. Another related limitation here is the potentially different mechanisms of loss of function observed in the pull down for suspected pathogenic VUSs. Measuring a variant's impact on binding ability and protein folding are two valuable metrics. However, it does raise questions about which one is more indicative of a loss of tumor suppressive activity, or if both are required to make a VUS pathogenic. The next steps here is to finish gathering my functional evidence for my pull down assays, and with my KNN classifier evidence, reevaluating the classification of VUSs of interest. The limitations that I've highlighted here confirm, though, that these results should be interpreted in the context of strong evidence types. So finally, I've actually included my third aim as a future direction here because it's answering sort of a different question than the first two aims, such that it's more of an investigation into the therapeutic applicability of a given variant. Thus, it does not need to meet necessarily ACMG guidelines to be of clinical benefit. Perhaps this could be a future direction in ways that we could make VUSs more clinically actionable in the clinical setting without having to go through the ACMG very stringent guidelines. And lastly, as a long-term future direction of this work, I'd like to integrate these models with ongoing pedigree analyses, which are being conducted involving Canadian families with observed BRCA1 VUSs. So the goal of this future direction is to validate the predictions and results from the in silico as well as the functional assays to gold standard results in the pedigree analysis. And the results of this project are important for a few reasons. Firstly, the BRCA1 specific classifier has shown to provide superior in silico evidence for classifying and stratifying VUSs in a certain domain of BRCA1. And the accompanying functional assays have potential for establishing strong evidence for classification of these VUSs, as well as their applicability as predictive biomarkers in the clinical setting. Each of these branches contribute to the overall goal of improving the interpretation of BRCA1 VUSs to more accurately inform clinicians as well as patients of cancer risks associated with VUSs and management steps afterwards. So we've made it to the end of the presentation. Thank you so much, everyone, for listening. I would like to thank my supervisors, Dr. Scott Davey and Dr. Harriet Filater, for their ongoing support, mentorship, and guidance throughout my master's. I'd also like to thank my fellow lab members, Nicole, Matt, and Kamran, for their dedication to the project, as well as their help and support along the way. My committee members, Dr. Mueller, Dr. Wang, as well as faculty members, Dr. Trishka and Natasha, for their help with feature selection. And finally, Dr. Greer for his leadership in the pathology department. So thank you, everyone.
So I'm sure Gabby would love to entertain questions. You first.
I have two questions. The first is, why did you only clone in the BRCT region rather than the entire BRCA gene? To see it like the VUS is like affect the entire thing.
That is a good question, Ty.
So can you repeat it for the
yes.
audience on Zoom then?
So one of the questions was why I only incorporated the BRCT sequence into my constructs when conducting my pull down assays, as opposed to incorporating the full length of BRCA1. So one reason for that is BRCA1 is, in relation to other proteins, quite a big protein. It's about 1,863 amino acids long. So for this given branch of the project, we are focused primarily on one aspect of BRCA1's ability. So bypassing some of the limitations of incorporating a larger protein into a plasmid, which could lead to potential complications down the line, we honed in to focus on just one aspect of BRCA1, which was the BRCT domain.
I said you think. What are the three like binders that you use? What do they do like mechanistically?
Like Abraxas, CTIP, and BOC1? Yep, that's a good question.
In the body, or those like Abraxas?
Let me pull back this slide here.
It's like the spinning primal. Oh, for them.
I can show back the spinning primal as well. That was one of my favorite slides, but I think that's perhaps one of the reasons why this kept shutting down. But anyway, so Abraxas, CTIP, and BLOC1, so they are phosphoproteins because they consistently have that phosphoserine phenylalanine motif that makes them common throughout those proteins, but they interact with BRCA1 to create those macro complexes. So you can see the top complex there is the interaction between Abraxas and BRCA1. The middle one, you can see the interaction with CTIP and BRCA1, and then BRIP, which is another equivalent to BOC1, is shown in the last complex on the right. So the majority of these complexes are used in mediator functions with BRCA mediator functions for homologous recombination. So the proteins themselves are actual proteins that are endogenously in cells that are involved in homologous recombination. They are involved in mediator functions. However, the BOC complex on the end there has been shown to be involved in actual effector functions in DNA repair.
So the BRCT is only the DNA repair. So I didn't try like the other. There's like 4 other mechanisms that like the chromatin regulation. That's called BRCA1, right? So that's not the piece.
So DNA repair is one of the more elucidated aspects of BRCA1. BRCA1 itself is quite an esoteric gene, and there's a lot of unknown about it, but DNA repair is one of the key drivers into it being a tumor suppressor gene. So that's why we focused on DNA repair for this project. And then BRCT, it has a established two binding partners that have been shown to be involved in homologous recombination.
I think that hand is coming up. Can you see it twitching?
Yes, Ivan.
I do have 2 questions. Could you go back to where you're comparing alpha, what's it called?
Alpha
missense,
missense, yeah.
and your model? The question I have about that is, there's obviously the difference of 6 mutations being misclassified. For the alpha missense, I know what it's looking for is for residue substitutions, which actually cause the protein to misfold, become insoluble, and whatnot. With your classifiers, with all the features that you were selecting, do you know if those are looking for this sort of misfolding, missense mutations, or is that part of the question about the residues that don't affect the structural integrity of BRCA1 but do affect the binding affinity? Is that included in those? Because if, say, those are included in your classifier but are not included in alpha missense, then maybe those 6 residues are actually interesting to see what those are, because maybe those 6 are the ones that that don't affect the structure but do affect the function.
Yeah.
Do you have any insight into it?
Yeah, I see what you're saying. I can bring up the actual list of in silico tools that were selected from the feature selection algorithm. And if it's all right, Ivan, if you could repeat the question part of it again.
Are they looking for structural permutations that affect the structure, unfolding of BRCA1, or the binding to its binding partners?
So the majority, or the top performing tools for BRCT in this example, the majority of them were the machine learning tools. So they were trained on large data sets, on known consequence variants. So they do like
what the consequence is. Is it the effect on the BRCT BRCA1 itself, or on the binding?
Sorry, can you repeat that question one more time?
So in the tools, you have the mutations that are predicted to be pathogenic. In your own experience too, you've noticed that the loss of binding could be either because of actual loss of binding or because of the loss of the structural integrity of BRCA1 itself.
Right.
So in those tools, is there any insight into which mode of pathogenicity is the one that you get in particular mutations?
That is a good question. I don't mean to dance around the question at all. I will highlight one aspect of variation between the ring domain and the BRCT domain, and that's alpha align GVGD, because that is a in silico tool that is based on the sequence conservation as well as biochemical impact of a variant. However, that wasn't one of the top performing tools for the BRCT domain. So perhaps it's focused on, or the features that were selected for BRCT veer farther away from those tools that assess primarily conservation and sequence conservation. But in terms of the types of tools and maybe what it's assessing, that is a good question. I don't have a specific answer
If it's
right now,
readily,
but
available to know from those tools already.
Mhm. Right.
Can I just add to that, because I think this is an interesting point of discussion that I remember from hearing your presentation last year and several other talks that are attempting to do this sort of thing, in particular people from Anna's lab. For a lot of us, it's just black box, and so not really understanding how the algorithms are working. But you did mention at the beginning of your answer that it's trained on large sets of data.
Mhm.
So whether it's actually extracting what you're talking about, information that's linked to a particular aspect of structure, or it's just seeing correlation, and maybe you don't
Mhm.
even need to know. Scott, do you have a comment on that?
So I would comment that based on your functional results, you know that your model has pulled out things that both just change the binding pocket and also change the structure of the protein. I think maybe what I was trying to ask is the ones that are different in the AI, are those localized to one or the other of those categories?
Mhm.
Which I think would be really interesting to look at. And have you looked at that?
I haven't looked at that as of yet, the specific variants that were misclassified, but I think that could be a future direction that would be interesting to look
Yeah,
at.
it's interesting, and it gets to this issue, you have all these different tools to use. And you're kind of, I don't know if it's just empirically finding out which ones give you the best result without really knowing the answer to questions about why a
Mhm.
particular one works better.
Can I add that the difference between algorithms and your classifier? So your classifiers, which are mentioned here, some of them they're using in their training, they're using clear data.
Mhm.
Whereas algorithms, they don't use clear data.
Right.
They don't train their model based on clear.
Mhm.
But your data is obtained from clear. It might be biased a little bit. So that's why you might have that higher accuracy.
Right.
David, I'll
Yeah,
let you
I had a
go.
question really just in follow up of Ivan's question. So. Maybe you want- do you want-
We can
Well,
circle
certainly-
back.
So
We got time. It's still light out, folks. Don't worry.
It's gonna take a while. Great presentation. David.
Thanks, Dr. Leckrell.
So, I think alpha missense uses a lot of information from AlphaFold.
Mhm.
correct? So that when it's looking at structural predictions, it could be that there's a lot of unstructured regions of BRCA1. And in fact, the data you showed us looked as though there's lots of unstructured regions of the protein. So is that why alpha missense is not doing so well in many of these predictions? Simply because
Yeah,
the structural evidence is uncertain.
Mhm. That is a good point, Dr. Lillicrop, and I think that could definitely be an outcome as to why alpha missense isn't performing as accurately, given that there are different structural components to be considered with BRCT. And it's not fully elucidated as of yet the impact of each structural component on BRCT's tumor suppressive ability. So that could definitely be an option.
I'm gonna let Shifa go next.
This is also kind of into the discussion. So for the some or like the variants of unknown significance that were misclassified in your KNN model. So when in the MATLAB learner, and you ran all of those different classification models, was it generally the same VUSs that were misclassified several times? Or...
You better repeat that, because I think.
Oh, yeah. Sorry, the question was, when assessing the performance of the KNN classifier, there were misclassifications of pathogenic variants as benign, and the question was whether those same variants were misclassified across other tools as well.
Yeah, the other classification.
So that's a very good question. I hadn't looked into the specifics into which VUS or which variants weren't or were misclassified, but perhaps that would give patterns into maybe why it's not as strong at predicting those ones as pathogenic.
Okay, well, You want to ask the second half of his question, then I'll
No,
get to
no,
you.
question. Can you go to the slide where you have the workflow for transactions and verifying the documentations? Sorry, not this one, the one after the where you had MDAB for.
The MD, yeah.
Oh, sorry, the
Yeah,
PARP inhibitor
yeah.
sensitivity. Yeah.
Yeah. So when you were describing the cell line, I don't remember how exactly you mentioned that, but on the slide it says BRCA1 minus.
Mhm.
So I'm just wondering what's hiding behind that minus. Because just out of curiosity, I looked up the cell line and it says that BRCA1 is mutated, but the way it's mutated is it has a single nucleotide insertion in exon 20 out of 22.
Mhm.
So it's a very C terminus of the protein that's mutated. So functionally, if you were to do a Western blot for BRCA1 in that cell line, will that protein be present there? It's just that its C terminus is going to be slightly functionally impaired, but the rest of the protein is all right? Or does it cause for the entire protein to, just be non-functional? Or just basically, and if whichever case it is, how does that affect your results if you are suddenly to put a second isoform of BRCA1 with a different mutation in? Because at that point, if you have two different mutations in different spots, could they just, one isoform be, compensating for the other one, or I don't know.
Right.
Just on the basic level, is there BRCA1 in that cell line at all?
So yeah, BRCA1 is in that cell line. So I would expect with a Western, we would be able to see it in the pull downs. However, it is mutated to an extent that it's confidently said that cell line has BRCA1 mutated. Unfortunately, we don't have the luxury of being able to work with like a fully BRCA1 knocked out cell line, given that it is like embryologically lethal if both BRCA1 copies are knocked out. So this was the closest that we were able to get to a BRCA1 like deficient cell line. However, there have been studies that have conducted similar PARP inhibitor sensitivity assays using MDA MB436 cells in the past. So that's what led to the development of this assay based on the success of previous ones.
I'm gonna go with Matt first.
Just two interesting points to add on to that in silico discussion, like two tools that were on that slide definitely stand out as making our Gabby's algorithm stand out from AlphaMissense. BRAS HDR is a machine learning algorithm based on the results of the HDR assay. So it's more functional than it actually is computational. And then UCRED2 can look at PDB structures of where proteins and ions interact and factor in a prediction on if it retains or loses binding into their pathogenicity prediction, which is beyond kind of the scope of what AlphaMissense is able to do.
Thanks, Matt. And can you see the chat too?
Oh, sorry, they
Yeah,
were.
because I was asking people on the Zoom if they could hear you. I don't know if they could hear you, Tashifa. I'm sorry, they didn't hear you. So you might have to repeat that. That was more a comment than a question, I guess.
Yeah, thanks, Matt.
Chris?
So you implied during your talk that you don't know which partner is actually responsible for the transforming the, right? So you said, CTIP or Abraxas wasn't clear. Isn't that known from pathogenic mutations in terms of which of those is actually the main contributor to pathogenicity?
So if one binding ability is lost, that would equate to loss of tumor suppressive ability, is what you're saying, Dr.
Yeah. Yeah, no, so don't we know which the part are, like, does the mutations affect the binding of both partners, or is there a selectivity there? That one partner is more or less important in terms of its binding?
I'll go back to the slide showing the So I think that is a very important question to ask. And I'm sure there is literature backing if there's certain interactions that are more pivotal for homologous recombination than others. As I've outlined, each complex is involved in its own sort of standalone, as well as overlapping roles in homologous recombination. So I would assume that variants that have lost binding ability to both Abraxas and CTIP, for example, the arginine to proline one, would be more detrimental than a variant that has only lost its ability to Abraxas, but has retained its ability to bind to CTIP as well. If I were to guess, if one binding activity is more detrimental, or the loss of a binding activity is more detrimental to others, I would assume that it's maybe one of the mediator functions, so maybe something like a BRCA1 to CTIP, as there are other mediators that bring repair mechanisms to sites of DNA damage. So maybe the Abraxas complex to the Abraxas binding to BRCA1 may not be as essential, given that there are.
Maybe the corollary to that is, are there mutations that are specific enough that you lose binding to one protein and not others?
From the pullout assays, or
Well, in general, yeah, in pull down assays.
we were able to see that there were
No,
some.
no, but in other people's studies with pathogenic mutations, is there a mutation that affects one like CTIP and not Abraxas? Or is there, can you even differentiate those? Or they find both?
Well, from my from literature reviews, a lot of the functional assays that are done to assess variant pathogenicity are done with homology-directed repair assays. So they look at BRCA1 as a total in terms of its ability to perform homologous recombination. This is a little bit different because it's looking at the actual functional activity. I'm sure there are assays out there that specifically hone in on BRCT, but the majority of the ones that I've looked at focus on BRCA1 as a whole, which is a little bit less of a mechanistic impact in terms of which binding activity is more important. However, we thought it would be important to focus on that functional activity to see maybe this could answer if there are some binding activities that are more important than others when we assess them through the pedigree analyses.
That just sort of for all of it. So you were saying, your flag assay, the levels of protein differ. Is it in fact a good assay? Is protein stability by itself predictive in this context?
So that is another, like, it seemed like a setback at the time that I wasn't able to achieve consistent flag banding across,
Right.
but it did lead to another insight into how these variants are actually affecting BRCT functionality. It was initially just to assess its binding ability, but now it looks like it can provide insight into the overall stability of BRCT as well. So it could be an assay that could be used as a double header almost, where it gives insight into two aspects of a variant's impact on BRCT. But I still do think that this is a good assay, and it does have limitations, but it also does show future directions that can be focused on with future assays to focus either on this protein stability or maybe focus more on the binding ability.
Just to add on to that, maybe you said this and I missed it, but is it possible to express conveniently recombinant full-length BRCA1?
We can.
You can do it. So you
yes.
could, in theory, try and do these analyses with a full-length protein, but you're going to a specific domain, and trying to do it that way.
Right. That's true.
But that also gets to this issue of the structure function that you're alluding to with the, what is it, the 436 cells and 463 cells. So, because I was going to ask that question too, I assumed it was a null mutation. Null mutations you can achieve with these, you can find these.
Last time I looked back, it was like a mutant BRCA1.
So it's some kind of a truncation or it's a frame shift, so you're presumably getting a heterologous sequence for most of the end of the protein. Okay. Because if you had a null cell line, that would presumably be ideal to do these sorts of rescue experiments. And because I think you were talking about doing rescue experiments with a cell line that still has a mutant BRCA. So that's going to be complicated.
Right.
Right.
And it also brings up the issue that we're doing this with transient expression as well. So we're not selecting our cells that have the BRCA1 that has successfully took up the vector as well, but we're basically just seeing if there is a difference.
For your pull down assays, that's fine. But I was thinking more, I think this idea of looking at a shift in drug toxicity is an elegant way of doing this. It might be quite quantitative, but for these sorts of DNA repair assays, are those assays convenient enough to do and get good quantitative data there too, or is it more binomial?
We do have the ACC 1937 line, which is a C-terminal truncation, null used. The reason we chose the other ones to depart facets is because other people could use them.
Okay. Yeah, because the part assay, I think, looks pretty interesting. I had one other question about the pedigree analysis, so.
Yeah.
How many affected family members do you have to get to before you determine from that analysis that it is a pathogenic mutation? Because you were talking about it, it's difficult because it takes time. And I assume it takes time because it takes a while for people to develop cancer or
Right.
to subsequent generations to develop cancer. So is that a practical way of assessing these mutations?
Well, in Ontario, due to the financial constraints, reflex testing isn't provided for people who have VUSs, but in terms of actively searching out for families who have BRCA1 VUSs, it is a future direction of our lab in collaboration with other clinical labs. But in terms of the amount of families that we would need, the number itself is escaping me right now. I'm not sure.
It's not a specific number of families, it's how many people, and it depends on the variant.
Yeah.
It's how many people are alive.
Yeah, it sounds pretty challenging.
It's hard.
And going to large cohorts where you presumably can find these US in large cohorts that have been brewing for some period of time, is that used as well?
I believe so.
Some of these are in one family, that's it.
Okay, so it's not practical,
It's
just,
population studies.
Family size to decrease.
it's, yeah.
Collaborators, better.
I don't see any other hands wagging and nothing from the chat, so join me in thanking Gabby for a wonderful job.
Thanks, everyone.
Good.